All right, so we are in the third section on muscle contraction and relaxation. And this section is a long section. We're going to talk about a lot of steps and details involved in the steps. And I want to spend time on this. I think this is, to me, the hardest part of chapter 10 is this video. So it's all, this section is just this whole video and nothing else that we're going to talk about but this in this section. So let's get started on how muscles contract. What even starts them off. So it all starts at a place called the neuron muscular junction. So where the neuron is going to meet the muscle. So we said nervous system's job is to communicate, right? That's its job, is to receive signals, send it up to CNS, decide what to do with it, and send it out to PNS to have an effector change. And so those nervous cells can meet up with other nerves. They can meet up with endocrine glands. They can meet up with muscles. So at the neuromuscular junction, that is where the neuron is coming down. So here's our neuron, and you can see the Schwann cell here with its myelin covering it up. And then it's going to come down at these little terminal buds. Right here. These are our little synaptic terminals. And so an action potential happens in the nerve. It gets triggered, it gets turned on, and action potential goes down the nerve, and then it's going to hit the muscle and create an action potential in the muscle and turn it on, so it's going to contract. So that happens at the neuromuscular junction. Let's go through the steps involved in this process. So we said an action potential is coming down the nerve cell. So it's coming down the it started up at the axon hillock, it's coming down the axon, it's going to go down to the terminal bulbs, and so action potential is come down. We got these arrows, come down. We are dipoling and sending a signal down the nerve to this synaptic bulb down here. And as this action potential comes down and arrives at the terminal, it's going to, because it's an electrical signal coming down this cell's membrane coming down, it's going to, since this electrical signal is going to turn on these voltage gated calcium channels. And we're going to talk about the electrical signal and what voltage gated means in just a little bit. We'll get into more detail on that. But right now we're just going to cover action potential happened to the nerve. I told the nerves in going from my CNS through my PNS to turn on these muscles and move this part of my body with a communication. That's what's happening here. So it's going down the nerve, and now it's going to meet up at the muscle. When it goes down to the nerve, it's going to open these voltage gated calcium channels. Remember, I told y'all calcium was so important, not just for bones, but for communication. So these voltage gated calcium channels get to the right voltage and they open, and calcium is going to start flooding in. So calcium starts pouring into the cytoplasm of this cell, of this neuron. And when the calcium comes in, it's going to cause this acetylcholine to be packaged up and start exiting out of the bulb. So action potential comes down, and we're opening up these calcium channels. We've packaged up this neurotransmitter. A neurotransmitter does not have to be acetylcholine, but when we're talking about skeletal system, we're talking about acetylcholine. But it could be dopamine or any other neurotransmitters out there. It's a chemical that sends a signal to the rest of your body. So in this case, it's acetylcholine, and the nerve cell has packaged up the acetylcholine in a little bitty vesicles, and it's going to exocytosis. It's going to send those outside of the nerve bulb, outside of this axon terminal. And so that now we're in step 3. Those acetylcholine is going to exit via exocytosis, and it's going to rupture, and acetylcholine, you may see it called ACH right here, these little green dots. ACH is going to diffuse across the space between the nerve and the muscle. That space between the nerve ending and the muscle is called the synaptic cleft. The synaptic cleft. So it's not a very big space, but it is a little bit of a gap. Synaptic cleft. between where one cell and the other cell meets. So it's going to leave the neuron, it's going to go across, and it's going to release all these chemicals, neurotransmitters called acetylcholine. They're going to go across the cleft. We're in step 4. Acetylcholine goes across the cleft, and it's going to bind to receptors on the sarcolemma. And remember, the sarcolemma is the cell membrane of the muscle cell. So now the signal has come from the nerve. We followed steps 1 all the way through to 4. The nerve sent out the acetylcholine across the synaptic cleft, and it binds to the muscle cell. So that's where we're at. We've gone through these steps. Triggering the nerve for an action potential, goes down, opens the voltage-gated calcium channels. That's going to trigger the acetylcholine that's been packaged up in there to rupture and leave the cell, exocytosis out of the cell, go across the cleft, and now that acetylcholine, the neurotransmitter, is going to bind onto the muscle cells on these little receptors on the outside of the muscle cells, on the outside of the sarcolemma. So that's where we're at. We're 4 steps into this. It is happening at the neuromuscular junction. One more time, test question. Neuromuscular junction is where this is happening, where the neuron meets the muscle. Okay. Remember way back when we were talking about cells and how we talked about transporting things in and out and diffusion, right? And we talked about passive transport was diffusion, it didn't take any energy. And then our example for active transport was the sodium, potassium pump. And we said that that did use energy because it is pumping things in and pumping things out against their gradient. And we used ATP to be able to do this. And what did we pump out? We were pumping out a whole bunch of sodiums. So we were pumping out 3 sodiums that we were pumping out of the cell. And then the next step was to bring in the potassiums. All right, 2 potassiums into the cell. And this takes energy, but this is what we want to do. This is what the cell wants to do. We are creating this electrochemical gradient by doing that, by pushing all the sodiums out and keeping all the potassiums in. We're changing our charge on what's inside the cell and what's outside of the cell. So that difference in charge overall outside of the cell is going to have roughly a positive charge. So we have this picture here that I stole from Pearson. They have the best images on this chapter. I'm trying to stick with just OpenStax, but these are too good and I want you to have them. So until OpenStax can get some great ones just like these, I'm going to keep them. So outside the cell, positive charge in general. We call this the resting membrane potential. By the way, when the cell is at rest, outside the cell is positive, and inside the cell, overall, despite us bringing in the 2 potassiums, and we're sticking all those, the 3 sodiums out there, overall, when we add up all the ions inside the cell, it is a net negative charge when we are resting, when the cell is just hanging out, doing its thing. So outside is positive, inside is negative, and there's a difference in charge. So if you were to connect these two, like a battery, you would see the difference in charge at a resting membrane potential is negative 70 millivolts. You might need to know that on the test. Negative 70 millivolts is the charge difference between the outside and the inside of the cell. That is called our resting membrane potential. And part of that potential is helped by that sodium potassium pump. There are also leaky channels, and those guys operate on simple diffusion, where we got all this potassium, and we put it inside, and that's what we're wanting to do. We're putting it and pushing it against the gradient and creating this lots of gradient inside of potassium, and some of it's going to sneak out these leaky channels. So there are leaky channels for not only potassium, but also sodium has leaky channels, so they can sneak in too. But our sodium potassium pump tries to make up this difference and keep sodiums out and potassiums in. Resting membrane potential. So this is normal when the cell is resting. The neuron is not triggered an action potential. We are just hanging out. This is again, resting membrane potential. But we've gone through the steps of the neurotransmitter going across the cleft. We had an action potential. The neuron is sending a signal. It had the voltage-gated calcium channels open up. And that told the vesicles inside that we need to release this acetylcholine. The acetylcholine went across the synaptic cleft. That was like step number 4, and binds to receptors on the sarcolemma, on the muscle cell. So we're at step 5 now. So step 5, that acetylcholine, abbreviated as ACH, it's these little green guys right here. Those went across the synaptic cleft, right? And they bound onto the protein receptors that are embedded into the muscle. What happens when those bind on? Well, when those bind on, we're going to reverse everything that we worked so hard with the sodium potassium pump, and we worked so hard to pump out the sodiums and bring in the potassiums. That gets reversed on an action potential when it gets triggered. So sodium is going to start flooding into the cell, and our potassiums start leaving. So when sodium starts flooding into the cell, flooding into the cell, flooding into the cell, remember, normally at resting, here's resting, and we were at negative 70 millivolts. But we are, and we, here's positive over here. Here's neutral at 0. If we're at negative 70 and sodium starts flooding in, all of these positive ions start coming into the cell. They're flooding in the cell. They're flooding in the cell. They're going to change our overall net charge, because before we were positive outside and negative inside. But we can't keep that negative charge if a whole bunch of sodium start flooding in on us. So we are going to flip it. We are now going to start going closer to 0, because all these positive sodiums are coming in. All these positive sodiums are coming in. And that's called dipole. It's called depolarization. These ions that are positively charged start flooding into the cell, and then they're going to change our overall charge. And we start going closer and closer to the positive end on the millivolts. So we are depoling. When this action potential comes down, triggers the synaptic cleft to have this acetylcholine that binds on, and now we are having local depolarization at the end plate where the neuron bulb, where the axon terminal is going to meet up with the muscle cell. So we are depoling. Again, we said we're getting, it was negative 70 down here, and we're getting closer and closer to positive numbers because of all those positive sodiums that are floating in because of the acetylcholine that opened up the channels. So sodium's flooding in, it's flooding in, sodium's flooding in, and that's going to change our charge. And now, instead of being before where we were positive and negative, now we're more positive on the inside and negative on the outside. That is going to cause a wave of depolarization to go down the cell. So it touches the cell, it touches the nerve comes, and the synaptic cleft sort of kind of touches, but not really. The neuron is going to touch the muscle cell, and it's going to set off those acetylcholine. It's going to open up the channels once the acetylcholine binds, and sodium floods in. Sodium floods in, it makes it positive, positive, positive. That's called depolarization. The cell's changing charge. And now we are going to have this wave of dipole that goes down the line of the muscle cell. It's going to go down the sarcolemma and travel down as long as that stimulus is coming, as long as the nerve gets stimulated, as long as an action potential is still happening, and as long as acetylcholine is still being released by that neuron, then it is going to travel across the cleft, and we are going to have dipole happening. Now, it's going down the sarcolemma. What happens when it gets further down? Well, if the charge is still going and depolarization is still happening, we are going to open these voltage-gated sodium channels. There's voltage-gated ones, so they have to be to a certain charge before they'll open up. If we're just resting, these bad boys are shut. But since I told y'all that our charge is getting more and more positive, then these voltage-gated sodium channels are opening, and more sodium comes in. So we are just getting up, 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 positive, positive, positive. And we are closing these voltage-gated potassium ones, and they shut. And this action potential is going to carry itself down the muscle cell. Again, as long as it's getting stimulated, as long as acetylcholine is coming across the cleft, we are going to depole and then send that action potential down the muscle cell. Stop it, Ace. So here it is traveling down the muscle cell, and we said that every muscle cell has these T-tubules, and we mentioned that they were these little holes where they travel down and they bump up against the SR in this terminal cisterna. So here is our action potential. It's coming across, and then it's going to go down the T-tubule, and here is our terminal cisterna of the SR. And remember we said one of the jobs of the terminal cisterna of the SR is to hold the calcium. And so the calcium was bound in there with that calsequestrin. Now is its time to shine. It's the time to shine. So the action potential is coming across the sarcolemma. It's going to go down into these little T-tubules, and it's going to hit the terminal cisterna of the SR at this triad. And then we are going to release the calcium throughout the cell. It's going to release the calcium. Calcium ions are released. So I said that this is going to keep going until the neuron stops sending its signal. It stops sending the acetylcholine, the neurotransmitter, from the neuron to the muscle cell. Once that stops happening, we will stop sending an action potential, or we can send in acetylcholinesterase, which is an enzyme that dissolves acetylcholine. And so either way, we are going to eliminate the acetylcholine and get that out of the situation. Once acetylcholine, yes, please. Once acetylcholine is out of the area, we have degraded it, eliminated it. Now we're going to stop it, because before we're at -70 at resting membrane potential. And remember, sodium flooded in. Sodium floods in, and that's going to bring our charge from -70 up, 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 up. And eventually we are going to lose that sodium. Those voltage gated channels are going to close, and because acetylcholine is stopped. And now we are left with repole. This is dipole. Over here is repole. So much better when the dog's not whining in the background. Okay, so we eliminated acetylcholine, which was our trigger, our neurotransmitter. Once we get rid of that, we're going to stop the sodium from coming in, and our charge is now going to stop climbing, and now we can repole and start to go back down. So that's repolarization, where our sodium is now going to flood out of the cell. We want sodium to come out on repole. So we are closing those voltage gated channels that was allowing the sodium in, and they're no longer allowed in. Sodium's not coming in those voltage gated channels. What is going to open? Potassium channels. So we are going to lose, I'm going to go back. We are going to lose, potassium is going to go out. Potassium is going to start flooding out. And what is potassium? It is also a positive ion, right? So when we lose that positive ion, it's going to drop our charge. So let's just change this guy to potassium too. Just all the potassium floods out at this certain voltage. And I can't remember what voltage it is. The last textbook had mentioned it. I want to say it was like 110 or something like that, but I don't think this textbook makes you memorize exactly what millivolt it is where sodium closes and potassium opens. So I'm not going to stress y'all about that, but do understand at a certain charge, it's going to go up, 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 up. And at that certain charge, we close, these shut down, and these open. And that's going to allow the potassium that are positive to start flooding out and bring our charge back down. Now we're here, dipole, and that was because sodium came in, repole, and that is going to have potassium go out, and that's going to drop our charge when we repolarize. I have these great videos that I did not make. They are Pearson's, but I think that they very much go in detail on the steps of the action potential. They're great study things to use to help reinforce these steps on what's happening when muscles are contracting. We're going to need to know the steps at the neuromuscular junction and how it travels across the muscle cell, goes down the T tubules, bumps up to the terminal cisterna in the SR, releases the calcium, and then we're going to talk about the sliding filament theory inside of the muscle and what happens there at excitation coupling. So here it is in a nutshell, and we're still not done yet with all the steps. It's a lot. So I got this whole video to go through it and all of these other smaller, other sub videos for you to watch because it's complicated. I know it's complicated, and I want you to hear it from multiple sources to help soak it in. This was like steps numbers 1 through 4. Action potential comes down the nerve. Calcium opens, neurotransmitters release via excitosis. Acetylcholine gets dumped into the synaptic cleft, meets up with the muscle. Then that muscle right here is going to undergo dipole because of the acetylcholine, and sodium is going to come flooding in, and it's going to dipole, and it's going to set off an action potential that sweeps across the sarcolemma down the holes of the T tubules, bumps up to the terminal cisterna of the, this is the SR right here, and it's going to release the calcium. So releasing the calcium. Now we're going to talk about the excitation, contraction, coupling, and sliding filament theory. So with the EC coupling, I just described all these steps. Here, I'm not going to say them one more time. We are down at this point where we have bumped up to the terminal cisterna of the SR. It is going to release the calcium. Calcium goes into the cytosol, the liquidy part inside of this muscle cell at excitation coupling. And then it's going to set off, here is another video from Pearson. Great videos, good ones. We are going to go into the sliding filament theory, the sliding filament model. I like Pearson's pictures. They go into detail better. So we're going to talk about the sliding filament model. What is happening? So at excitation coupling, calcium gets released. That's where we left off. Calcium comes out of the terminal cisterna, goes into the cytoplasm, and then what? Then what happens? These are the steps that we need to know for sliding filament theory. So on the step 1, the calcium is going to come down, and they get near the filaments. Our filaments, remember, myosin was the thick stuff and actin was the thin stuff, and then they would come together and they would overlap. But what makes them do that? We're going to talk about that. That's the sliding filament theory. So when they slide together, and then when the muscles relax and they contract again, that's sliding filament. So the first part, calcium comes in and says, you know, I'm here to everybody. Then calcium is going to bind onto, remember we talked about the little brown parts. So there was troponin, this little guy, on the tropomyosin. Calcium's going to bind to these bad boys. And when it binds to the troponin that's stuck on the tropomyosin, this stuff, it pulls away the string, the tropomyosin. It's gonna peel away the tropomyosin. It's gonna peel it all back and expose those binding sites on the actin. So now you can see, look, we got active site here, we got an active site here, there's one here. You can see all these active sites that get exposed because the calcium's there. Because calcium came out of the terminal cisterna, went into the cytoplasm, bound to the troponin, pulls the troponin and the tropomyosin away and exposes these openings. So active sites are exposed. Active sites are exposed. Now we can put the heads in, put the male parts and the female parts. The heads are going to bind onto the active binding sites on the actin. So that's called a cross bridge. Cross bridges form. And remember I told you that the head of a myosin filament is movable. So right now it's cocked back, and when it binds, it's going to form a cross bridge, and then it's going to move. It's the power stroke is what they call it, when the head moves and pivots, and that's going to physically slide the guys together. The actin and myosin are going to slide a little bit closer every time this happens. So we formed a cross bridge where the myosin head fit into the actin binding sites, and then it did the power stroke where it pushes across. And when that happens, it's going to also get rid of this old ATP that is now ADP. It's going to get rid of that. So it formed the cross bridge. It did the power stroke where it's going to pivot. And that old ATP that is now ADP, the old ADP goes bye-bye, and we don't need it anymore. We're going to get another one that's going to come along and help us out. So we did the power stroke, we lost our ADP, and we are going to now detach our cross bridge because it did its job. It moved the filaments like we needed it to. And then as this guy is detached, we are going to put on another ATP, and that ATP is going to break off the third phosphate. When we break off the third phosphate, that's how we said that we are, ATPs are energy currency. When we snap off that third phosphate, we use that energy for something. We're powering the sodium potassium pump. And in this case, we're using that energy to cock that head back into position. So it was relaxed. Now we got to put it back, and put it back there. It takes energy to do it. So now we've used our brand new ATP, and now it's ADP, and the myosin head is cocked and ready to go again. It is reactivated. This is the sliding filament theory. So let's go back through all these guys one more time. Action potential came across the sarcolemma, goes down the T tubules, bumps up into the triad. That's the T tubules and the terminal cisterna of the SR. That is going to tell the SR, release the calcium. Calcium goes into all the cytoplasm and all over the cell, because normally calcium's not hanging out in the cell. It's stuck up in the terminal cisterna, but we've released it. When the calcium gets released, it's going to bind on to the troponin, the little brown pieces, and that troponin is going to, with the calcium, move out of the way. It's going to pull that tryptomyosin, these stringy bits, out of the way, expose those active binding sites. We've exposed the binding sites, and then the heads can now bind onto the binding sites. So the myosin heads bind onto the actin, and that forms a cross bridge. And remember that myosin head is cocked and ready to go. Once it forms the cross bridge, it can now trigger itself and actually slide the filaments around. So we are going to have the power stroke where the filaments actually slide. They get, a little bit closer on this contraction. And then that old ATP, the old ADP goes away. And then we pivoted, ADP goes away. We don't need it anymore. We did the job that we were supposed to do. We slid the filament with a power stroke, and we're going to detach our cross bridge, and we're going to put on a new ATP. We're going to use that new ATP to now cock the head of the myosin and get it ready to do this all over again. So it's reactivated. This is happening over and over and over again. And remember, those thick myosin filaments have heads all over the place. Here's one, here's one, here's one, here's one, here's one, here's one. So this is happening constantly. As long as that calcium is in the area, all of those little heads are coming together, and they're making the muscle contract. That's the whole point of this to happen. So that is the sliding filament theory steps. You will probably need to put those in order on the lecture test. So let me know if you have questions about the sliding filament theory steps. They're also in your chapter outline, and I am sure they are on those Pearson videos that are very in-depth and very good videos. So at rest, the actin and the myosin are just barely touching, right? Because we said that there was this I band and there was an H band, and the I band and the A band, and the A band was dark and the I band was light colored. So this is at rest. And then when we trigger our muscle contractions, they're going to come together and overlap more and more and more and more. So where that light area, that eye band, is much, much smaller, and the muscles, it's contracting. The fibers are getting closer together. The muscle is shortening up as it does this. This is muscle contraction. So if you were looking at it under the microscope, here it is at rest. And you look at this, you got this big old space right here, big space between the I's and the A's. And then over here, where we are contracted, there's not a lot of gap. So here's our sarcomere. And we have this huge space. And here it's much closer because everything has come together when it's contracting. So it's much darker. There's very little light space in between these dark zones. This is the sliding filament model. This has all the steps from sending this action potential down the neuron, down those axon terminal bulbs to meet up at the neuromuscular junction, to send the acetylcholine across, and it's going to depole the muscle cell. It's going to send an action potential down the muscle cell. The sweeping depolarization goes down the muscle cell, down the T-tubules, setting off the SR to release the calcium. Sliding filament theory steps come into play, and the muscle starts to contract. And as long as it's getting the signal from the nerve and it's sending out acetylcholine, it's going to continue to contract. And then once the nerve stops sending acetylcholine and acetylcholine gets cleared out of the situation, we're going to go back and repole, and we're going to stop contracting, and the muscle will then relax and go back to normal. So when we repole, we get rid of the acetylcholine. When we're repoling and we don't get that signal anymore, then those calciums are going to release off of the troponins. They're going to go back into the SR, and everything will go back to resting. So we need to know these steps. I know it is a lot. It is complicated. Look at your chapter outlines. Watch the videos. Look at these images on the PowerPoints. It's a lot of stuff. It's a lot of steps. Please email me if you have questions. Here's that last part with the cross bridge. Sliding filament theory is here on part 3 of this video. The last part of this video, the last part of this section that OpenStax just kind of sticks it in here at the end, is muscle metabolism. And so that kind of leads us into the next section. So there's different forms of energy that our muscles can use. And when we are contracting, we'll call that a twitch. And so how many twitches can you get with the different energy sources? So our muscles are going to store different things. We can store ATP. There's some free ATP hanging out. There's not a whole lot of it, but there is some ATP hanging out in your skeletal muscles. And so those guys, if we wanted to look at ATP and how many we can get, here's our free ATP. And it says about 2 seconds worth or 10 twitches that you can get using free ATP found in your muscle cells. We can also take that ATP and combine it with creatine. Thank you. And that is going to then be able to make a little more. And if we use creatine, we have it as abbreviated as CP here. That's going to give us a little more twitches. And instead of 2 seconds, we get 15 seconds worth of work if we combine creatine with the ADP and create our ATP. So a little bit more twitches, a little bit more work that we can use if we're using creatine phosphate with an ADP to create creatine and then ATP. If we use glucose, we are going to store glucose. We mentioned this before, that glucose is our energy molecule. It's our energy currency molecule. And if we don't use it, then we're going to store it away as a polysaccharide for animals called glycogen. We were going to store glycogen. So if we store glycogen away in our liver and in our skeletal muscles, we can use that. And so we can use glucose if we break down, if blood sugar is low, if we need energy levels are low, we're going to break apart glycogen and release the glucose into the bloodstream so we can use it. So if we were using glucose, there's 2 different ways we can break it down. We haven't gone into cellular respiration in depth yet. I think we probably go through that, I want to say, in AP2. But with cellular respiration, there's aerobic and there's anaerobic. The first part of cellular respiration is anaerobic, meaning it doesn't need oxygen to use it. So if we were using glucose, if we were breaking down that glycogen to get glucose, glycolysis, um, we get about 130 seconds if we're functioning anaerobically. What do I mean when you're functioning anaerobically? You are working out really hard. You are working out those muscles so hard that they are starting to get tired because they are using up all the available oxygen. You are in, that heart range. If you're using your watch to monitor your heart rate, you're in that aerobic zone. You are using up a lot of energy. And if you are, I don't know, lifting weights, or I like to hike, so I'm hiking up the mountain and I'm trying to truck it up there, my quads are burning and they are using up the available oxygen, then I start to go anaerobic. So I am in this phase here. If you're working out really, really hard, your muscles are pulling in all the oxygen, then they're starting to use it up and you start to go anaerobic. And so if you're anaerobic and breaking down glucose, you get about 130 seconds worth of work and 670 twitches. So it's way better than just that one free ATP. And we're going to get even better. So say you were working out, but you managed to stay in that nice, happy zone where you're not pushing so hard that you're pulling out all the oxygen and you are just, you just can't. If you can maintain aerobic exercise where it's still challenging, but you still get in the oxygen with aerobic metabolism breaking down glucose, then you get 40 minutes worth of exercise off of that, 12,000 twitches. So there's lots of different energy sources. Glucose put into glycogen, and that's our, our short-term energy storage in our liver and our skeletal muscles. And we can break down that glycogen back down into glucose and use it. And are we aerobic or anaerobic? If we're aerobic, score for us, we get a lot more energy that way. If we're working out so hard that we're going anaerobic, we're not getting as much. And then we get smaller and smaller. If we use creatine paired up with ADP to make an ATP, we get about 15 seconds. And then any of the free ATPs that were in there give us about 2 seconds. So different energy sources for us to power our muscles in the form of twitches. How many twitches do you get to make with that energy source? So we have some graphics here talking about the different energy sources. If we're using creatine phosphate and we're going to create ATP and creatine, are we going aerobic or are we going anaerobic? If we are doing really brief, short runs where we just need a real quick burst of activity, then we can run on anaerobic. We could do creatine, phosphate, and ATP and get those really short bursts. If you're doing the 100-yard dash or whatever, those kind of things, really quick bursts. If you are trying to run a marathon, though, then that is going to be different energy sources. You are going to need to carbo load and put the glycogen in your system so it's ready to be broken down later. Same images, same kind of thing. If oxygen's available, you are aerobic. If oxygen is insufficient, your glucose turns into pyruvate every time. That's going to be our glycolysis where we're producing 2 ATP when that happens. But our pyruvate, instead of going into the mitochondria for aerobic respiration, step 2 and step 3 of cellular respiration, when oxygen is insufficient, because we've worked out so hard, and my quads have pulled all the oxygen around if it's available, that pyruvate then changes and it turns into lactate. So it turns into lactic acid that starts to accumulate in your muscles, and that can lead to muscle fatigue, where your muscles just can't do it. They just get tired and you just can't, lift that weight one more time. You just can't do one more squat, whatever it is. And then later when you're recovering, after a while, your liver can then take that lactate, turn it back to pyruvate, and then we can use that again in the cycle for energy. So that is a lot in this video. It's a lot of steps. Please watch the other videos from Anatomy Zone and Pearson. watch the steps. You will have questions about the steps. You might need to put the sliding filament theory steps in order. You will. You will. I always ask that one. So please let me know if you have questions about the steps on muscle contraction all the way through.